Hello children, welcome to Vivid Botany. We have a question about this uh, opening of the DNA helix. Yes, it is a very good topic because without opening, a DNA cannot make out another DNA or a DNA cannot make out another RNA. We know that DNA has got two functions. One is autocatalysis, another is heterocatalysis. So this question revolves around the two functions of the DNA. In autocatalysis, DNA gives rise to another DNA. In heterocatalysis, DNA gives rise to the RNA. Now, if you see the autocatalysis where DNA gives rise to other DNA, so this formation of DNA from another DNA is called DNA replication. DNA replication. So, if you understand the DNA replication properly and you can understand the functionalities of DNA ligase and DNA polymerase, because the two you should know before attempting the question very clearly. So, I would like to explain about them as much as I can. So, DNA ligase is that. Ligase means uniting. So, DNA ligase is that which can join any two DNA fragments. Yes, any two DNA fragments by binding with ester bonds. We know ester bonds are present in between the phosphate molecule and the sugar, that is deoxyribose sugar. Generally, third carbon, fifth carbon of deoxyribose sugar are involved with ester bond with phosphate. So this ester bond of phosphate with DNA, deoxyribose sugar, is catalyzed by the enzyme DNA ligase. It's true, but you should remember that you cannot bring two separate DNA strands and can join them with DNA ligase. The problem here is that if two DNA strands are having not complementary nucleotides, you cannot let them to join with each other. That is the case here. So provided that the complementary nucleotides, that is DNA should have complementary nucleotides, then only the two DNA strands can be joined by DNA ligase by attaching the phosphodiester bonds. Next comes DNA polymerase. DNA polymerase is that which attaches the complementary nucleotides, that is in complementary with the mother strand of the DNA. So that is the case here. We should understand. In DNA replication, if you see, the DNA two strands get separated. That is the first and foremost function, and the two old strands act as here template. The two old strands act as here template, to which again a new strand of DNA is formed by adding the complementary nucleotides. For example, if the mother strand is having here cytosine, then the new strand will have guanosine to cytosine, adenine to thymine, thymine to adenine. Like that, the new strand is formed by DNA polymerase with a rule of complementarity to that of the mother strand. Yes, here the rule of DNA polymerase is that it can polymerize only in 5 to 3 direction. That is the case here. It can polymerize only in 5 to 3 direction, but not in 3 to 5 direction. This is wrong. You should understand it. So, what happens during the DNA replication? One strand is formed continuously called leading strand, the other is formed discontinuously, that is called as lagging strand. So, in lagging strand, the separated DNA fragments that are formed are called as Okazaki fragments. These Okazaki fragments are united with the help of the DNA ligase, just by attaching, by forming the ester bonds in between phosphate and sugar that forms the backbone of the DNA. So there lies the role of DNA ligase. Do remember, DNA polymerase will bring the complementary nucleotides to the site of this one, DNA, whereas DNA ligase will attach the Okazaki fragments or the discontinuous fragments of that DNA. That is the case you should understand the difference in between DNA polymerase and DNA ligase. DNA polymerase concerned with polymerization of complementary nucleotides with a rule of complementarity to that of the mother strand. DNA ligase is that which can attach any two DNA strands that by forming ester bonds in between phosphate and sugar molecule. And DNA helicase. DNA helicase is that it will unwind the double-stranded DNA. We know that DNA will be in coiled fashion. It is right-handed coil, especially the beta DNA or bDNA in secondary structure will coil towards right hand side or clockwise direction. So when it is in coiled manner, when it is in completely turned manner, it cannot undergo the replication. So I told you that the first step in replication is to uncoil the two strands of dsDNA, double stranded DNA. So who will uncoil this to separate who will separate the two strands of DNA? This responsibility is taken by the DNA helicase. So this DNA helicase will uncoil or unwind the two separate strands of the DNA. But what happens here is that after uncoiling the DNA, again the two DNA strands may get recoiled. To prevent the recoiling of the DNA strand, another enzyme is used called as DNA gyrase. Gyrase this is or topoisomerases. They prevent the further winding of the DNA double-stranded DNA strand. Right now, that is DNA helicase. Now comes to the RNA polymerase. RNA polymerase it is polymerizes the formation of RNA from the DNA. It's also called DNA dependent RNA polymerase. But here also for the formation of RNA from DNA, DNA needs to be uncoiled. But who will uncoil the DNA? That is the question here being asked in the NEET. It's not helicase, do remember. Helicases, gyrases, topoisomerases are involved only in DNA replication but not in RNA formation. Then who will uncoil the DNA? You know, uncoiling of DNA is compulsory for formation of RNA from DNA. Without uncoiling of DNA, that RNA cannot form from that DNA. So who will uncoil the DNA? Yes, that is the matter of point here. It is RNA polymerase which will uncoil the DNA. It's not DNA helicase, it's RNA polymerase. Do remember, it is RNA polymerase that will unwind or uncoil the DNA. Then the two DNA strands are separated. Then one of the DNA strand acts as template to which an RNA is formed with the polymerization of RNA polymerase, that is DNA dependent RNA polymerase. Here three factors are there in fact. One is the starting is done by initiation factor or sigma factor. This is asked once in NEET. Termination factor or rho factor responsible for termination of the DNA transcription. Elongation factor, it is a continuously it catalyzes the formation of RNA from the DNA. So the three factors are responsible for formation of RNA from the DNA. So here the important thing you need to remember in mind is that the initiation factor or sigma factor, termination factor or the rho factor, the two factors are transiently attached to the RNA polymerase. Transiently means loosely attached to the RNA polymerase. What do you mean by the loosely attached? See, for example, it's not long-lived, it is short-lived. For example, we have boarded a bus. The bus is completely filled up, but for us it is compulsory to board the bus and to reach our destination within time. So, though the bus is completely filled up, we board the bus and very hardly we may get a seat or somebody we call a tip of the seat, like an edge of the seat. We may somehow manages to sit there, but it's not permanent that we can't sit on that seat long time like that. So that kind of sitting is called as transient, which is loosely bound, which is ephemeral, short-lived. So sigma factor, rho factor both are transiently attached to RNA polymerase, whereas elongation factor itself is RNA polymerase itself acts as the elongation factor during the transcription of RNA from the DNA. So do remember that RNA polymerase will uncoil the DNA. Apart from that, RNA polymerase also polymerizes the nucleotides to that of DNA during the transcription. So let us see the question here. Name the enzyme that facilitates opening of DNA helix during transcription. DNA ligase? No, it cannot open the DNA helix. It is wrong. And DNA helicases, it opens a DNA but not during transcription, during the DNA replication. So this question is very much twisted. One should have a very good knowledge about the text of the NCERT. DNA polymerase. DNA polymerase will polymerize, synthesizes the another new strand of DNA in complementary with old strand of DNA. RNA polymerase is very much right because it is associated with transcription. See here, if one doesn't know the answer RNA polymerase also, if one does not know the answer of RNA polymerase also, you can do it right because RNA polymerase is the only one which is associated with transcription. Rest of them like DNA ligase, DNA helicase, DNA polymerase are not related with transcription. So one can directly put it, if you are having minimum knowledge that DNA polymerase causes DNA replication, RNA polymerase causes RNA replication. So if you have that knowledge, you can do this bit without any difficulty, without any flaw. So hope my explanation is reaching to you. You are able to understand it without any difficulty. So I think you can reach to a target, right, without any hindrance or like that. So hope this video is helpful to you, and please subscribe my channel Vivid Botany. We are discussing all the questions from Vivid Botany. Subscribe this channel. Thank you.